A concert hall. The audience hears beautiful music, a mix of violin, cello, piano, and oboe. Each microphone in the hall picks up a different blend of all 4 instruments. You have the recordings, but you want to separate the individual instruments. The musicians are hidden. The music is observed. This is the latent variable problem, and it appears everywhere in biology, from gene expression to multi-omics integration. The core idea: observed variables are combinations of fewer hidden factors. 20,000 genes are observed, but they're driven by perhaps 50 underlying biological programs: cell cycle, immune activation, metabolic state, differentiation. The model: X equals lambda times F plus noise. X is the gene expression matrix. F is the matrix of latent factors, the hidden musicians. Lambda is the loadings matrix, how much each gene responds to each factor. Genes in the same pathway load on the same factor because they respond to the same biological signal. Factor analysis estimates lambda and F from the observed covariance structure. If genes A, B, and C covary strongly, they probably share a latent factor. The loadings tell you which genes belong to which factor. The factor scores tell you how active each factor is in each cell or sample. Rotation, like Vermax, adjusts the factors to maximize interpretability. Each gene loads strongly on one factor and weakly on others, making the factors easier to name as biological programs. ICA is often confused with factor analysis. Both find latent structure, but PIGA extracts components that maximize variance, order from most to least variable. Factor analysis models the data as latent factors plus noise and estimates the noise separately. Who components are orthogonal by construction. Factor analysis factors can be correlated. For single-cell RNA-seq, PISDA is used as a preprocessing step, fast and orthogonal. Factor analysis is used when you want a generative model of how the data was produced. Independent component analysis takes a different approach. ICA finds uncorrelated components, their covariance is 0. ICA finds statistically independent components, they share no information of any kind. This is the cocktail party problem. 3 microphones in a room with 3 speakers. ICA separates the mixed recordings into the original voices. In bulk RNA-seq, a sample's expression is a mix of cell types. ICA can separate the independent cell type signatures from the mixed signal, deconvolution, without single-cell data. Non-negative matrix factorization constrains both the loadings and the factors to be non-negative. No gene can have negative expression, and no factor can subtract. The result is a parts-based decomposition. Each factor represents an additive biological program, a metagene. NMF metagenes are often more interpretable than PICA components because they correspond to coherent sets of co-expressed genes without the sign ambiguity that makes PICA components hard to interpret. NMF is widely used for topic modeling in text and for discovery expression programs in cancer. Latent variables, hidden factors driving observed data. Factor analysis, estimate loadings and factors from covariance. The maximum variance, orthogonal, fast. ICA, statistically independent sources. NMF, non-negative, parts-based. All assume linear mixing. Next, what if the orchestra is non-linear?